Hi, I'm David and this is the Biology Classroom. In this video, I'm going to teach you how to make perfect biological drawings for paper 3. There are two types of drawings you need to make in paper 3. The first one is a plan diagram. It shows the distribution of tissue in a specimen. The second one is a detailed drawing. It shows cells and all the visible structures. Let's say you are given a slide or photomicrograph of a dicot leaf. This is how the plan diagram should look like. You have to use lines to show the different types of tissue that you can see in the specimen and their relative thicknesses. Don't worry if it's a weird looking specimen that you have never seen before. It might happen. Just use the shape, size and color of the cells to decide if they are the same type of tissue. Normally, there's only one or very few labels needed, so it's okay if you do not know the name of all the tissue in the specimen. Now I'm going to tell you the criteria to fulfill if you want to get full marks for your plan diagram. Number 1 is the quality of your drawing. Make sure all the lines are clear, sharp, fine, unbroken, and not ruled. Do not shade any of the layers no matter how darkly stained they are. Since this is a plan drawing, do not draw any cell at all. Number 2, the size of your drawing. You should use up more space provided. In another word, make your drawing as big as possible. In a marking scheme, there's usually 1 mark given for the measurement of your drawing. As long as you exceed the minimum measurement, you will get this mark. Number 3, the number of layers in your diagram. You should use low power objective lens while drawing a plan diagram, but you can always use the high power lens to check and differentiate the tissue. Of course, the best thing to do is to identify the tissue layers precisely, but it can be very difficult, especially when it's something that you have never seen before. If you are not sure whether an area consists of one or more than one type of tissue, go for the latter. According to the marking schemes, if you want to get this point, you usually have to draw more than a certain number of layers, so just draw more. Number 4, the relative thicknesses of layers. Use the eyepiece reticule or a ruler to compare the thickness of each layer. Then, show it in your drawing. Number 5, labels. Always use a ruled label line. One label line is only for one name. Do not have 2 label lines shared by one name. Make sure your label line is in between the 2 lines represent the layer. Use a bracket if you have to. Only label whatever the question asks you to. Do not make unnecessary labeling. Sometimes the question don't even ask for labeling, just don't do it. This is a photomicrograph of blood cells. Let's say you are asked to draw 1 white blood cell. This is how the drawing should look like. So, how do you get full marks? First, the quality of your drawing. Again, all the lines should be clear, sharp, fine, unbroken, and not ruled. Feathery lines and dashed lines are not allowed, so do not make a sketchy drawing. Number 2, follow the instructions given. The question usually specifies the number of cells you need to draw. Do not draw more or less than the number. If you see the words adjacent cells, make sure the cells are touching, but they are not supposed to be overlapping. If you are drawing 2 adjacent plant cells, always check that you have 3 lines where the cells are touching. The middle line represents middle lamella. Number 3, the size of your drawing. You must use up most of the space provided. In the marking scheme, there is usually a minimum measurement your drawing has to exceed in order to get the mark. Number 4, show variations when you are drawing a few cells of the same type. It is impossible for the cells to be identical in real life, so in your drawing, show the difference between them in terms of shape, size, or orientation if you can see it. Number 5, the relative size of different structures. For example, if the nucleus of a cell is taking up more than half of the cytoplasm, you should show it in your drawing. Number 6, label only when you are asked to. Use a ruled label line and make sure it touches the structure you are labeling. Strictly use one line to one name. You can use bracket, but make sure it touches the structure as well. If you like this video, don't forget to comment, like and subscribe. You can also follow me on Facebook. See you again soon.